Hello, my dear friends. Best greetings from Kyiv, the capital of Ukraine. My name is Viktor Fursov. I am a research entomologist. I am beekeeper and teacher. I'm studying small parasitic wasps, which are pretty invisible in the world around us because they're pretty small. They're small from the size of 0.5 millimeters up to 2 millimeters. They're here on this poster. Here they're pretty big, but in original size, as I said, they're quite small. So that's why you can hardly see them around you. Only devoted entomologists and lovers of insects, professional and amateurs, can observe them in the nature, on plants, on seeds, inside their so-called hosts. Host, it can be host plant or host animal. This is organism which are useful as a meal for development of larvae of these parasitic and phytophagous wasps. Phytophagous wasps because their larvae are feeding inside plants, and parasitic because their larvae are feeding on the body or inside the body of different and other organisms, insects and some arthropods like Ixodidae ticks, for instance, were also parasitized by these interesting parasitic wasps. And not so many people have seen them. Even in our beautiful collection in our institute, we have only few individuals, few specimens of parasitoids of Ixodidae mites, because they're pretty small, just only 1 millimeter. It needs a great attention to observe insects, to observe Ixodidae mites. These ticks are just not very pleasant for investigation. Many people are afraid of them, but devoted entomologists can collect one type of insect, which is the host, and just wait for a very precise study of biology, behavior, and devoted entomologist, parasitologist can find parasitoid, very tiny small parasitoid which is developing inside the body of host or on the body of host, or just searching for the host to lay egg on the body or inside the body. That's why parasitoid causing the death of its host. So this is a difference between parasite and not parasite, but parasitoid. Some mites also considered to be parasitoids because they're feeding on other arthropods and can cause them death as well. But majority will be talking about parasitic wasps, which are indicated on this poster. So far so good. Let's start from the beginning about collecting of parasitic wasps. Yesterday I was talking about different effective methods, how to collect, how to gather material of this tiny plankton all around you in a summertime, in a spring, summer, autumn time, and study them under the microscope in winter time. I was talking about plants, how to use emergence traps or emergence bags just to rear insects and to get many material of these wonderful tiny small parasitoids. And here I want to show you, for instance, the book which is named Insects and Thistles. Thistles, these are plants of a family Compositae, very common everywhere, just like a weed. And you see here on this cover, you can see different insects: moth, butterfly, fly, and tiny bug, this beetle. So they're all feeding on this plant, on a thistle, very common weed. Everywhere, just in rural area, in agricultural area, in urban areas, everywhere, just thistles are very widely distributed. So all these are phytophagous insects. And here the book, which is written by Margaret Redfern in Britain. Here with identification keys and for photos of different groups of insects. You see several species of moths are developing on thistles. Several species of Tephritide flies, many are developing on thistles. Actually, thistle is a just traditional plant in Britain, and actually it's indicated even on a coin of £1 of Britain. If you know just about something about coins, then you have seen them. So this is national symbol. And also some other insects like beetles, Cassidine, Cassida vulgaris and others, some Diptera, some bugs, some aphids, some another beetles. So many insects developing just on one plant or just on a similar species of plant of this same genus, which is in English it's called thistles. Some species are quite close of a family Composita. So these are association of insects developing on thistle. And so each of these insects has a separate, very specialized parasitoid. One species is specialized on moths, another just on beetles, just especially on Cassidida beetles, which is some parasitoids of a family Eolophidae developing on larvae of Cassidina beetles. This so interesting flattened shape. This green beetle. I show you close distance. Cassida. This green beetle. On larvae, on pupae, and on eggs of these beetles, and some others developing inside the pupariums of moths, or not moths, of flies, of Tephoretidae flies inside flower pods. Just some beetles, like Curculanidae beetles, are developing also inside these flowers. So you see many phytophagous insects developing just on one species of weed, and many parasitoids are associated with the same phytophagous insect, and it's quite interesting to understand and to discover this miraculous question, which host and which is parasitoid, because parasitoid is usually associated with precisely very special host. There are some oligophagous and polyphagous parasitoids. Some parasitoids they like different hosts, several species, but some parasitoids prefer only monophagous, only one species of host. So very, very precise identification of host because of pheromones, because of sensors, because of antennae, just searching for only one species of host with very special biology and maybe hidden, such a hidden behavior living inside concealed environment, like inside the stems, inside leaves, inside roots, inside galls. So it's quite interesting to discover this miracle, to decide to solve the question who is the host, who is the parasitoid, and to identify the name of parasitoid. For that you need to use rearing, collecting plants, collecting phytophagous insects and separating them in small glass tubes and taking each sample in separate tube. Here some tubes with a cork, with plastic cork, because this is with alcohol. But for rearing, for breeding, you need to have empty glasses without corks. So cork must be removed, and instead of the plastic cork, you must use just the cotton, to close it with cotton and put inside the sample of phytophagous insect or maybe parasitoid. Maybe put here a little bit honey for parasitoid. Some put here inside small piece of plant for feeding phytophagous insects. And inside these glasses, small jars, or just bigger size, like glass tubes, you can put inside bigger leaf for feeding your insect. So insect will be growing, developing little bit, certain period of time, and parasitoid will be growing also on this phytophagous insect, and you can receive just precisely rearing from precise host. So you can say this parasitoid belonging to this, and only maybe on this you can suggest that, or you can observe precisely the observation through your observation that this species developed on the body of this larva or inside the body of this larva, because you can observe it under the microscope and you separate it different samples of different hosts separately in glasses. So not putting just a big plant inside cage with many parasitoid phytophagos, moths, dipteroflies, beetles, all together, and they're just crawling around all together, and so you cannot understand which host and which is the parasitoid. If you can separate them one by one, be carefully, you can observe them and solve this question. In some cases, it's important also to make dissection under the microscope with very tiny pins, the body of host, and it's important to make dissection. You can observe the larva inside the larva of host, or larva inside the egg of host. So very tiny, very precise, very observative and very delicate observations, but which can give just unique information about biology of host and biology of parasitoid, and precise solution of whether this parasitoid developed is precisely inside this host and precisely in the larva or in the egg, and you can receive either the egg or larva, and you can see different larvae inside the body of different hosts. And not many people can do it because it needs very careful observations, very precise fingers, like surgery, like medical surgery, just for cutting, for making very careful preparations, like doctor, under the microscope, to dissect the larva, to dissect caterpillar, the larva of mosquito, larva of beetle, larva of caterpillar, or caterpillar, or of beetle, or the bug, and look inside, making anatomical dissection, and finding, oh, this is egg of parasitoid, this is larva of parasitoid. These are different stages of larva of parasitoid. You can try to do it in the spring and summertime, and you can receive very interesting discoveries. Maybe not many people knew about larvae in this host and this host, or you can observe it with first. You can make photos, can make video, and you can share your knowledge with specialists which can use it maybe for scientific identification and for description of biology. You can make consulting with professionals like professionals, entomologists who are studying biological control in parasitoids in our department of taxonomy of entomophagous insects and ecological principles of biological control, where I'm working at the Institute, Schmalgausen Institute of Zoology of National Academy of Sciences of Ukraine in Kyiv, the capital of Ukraine, by the way. So you can make consulting if you like. You can use my email for contacts if you visited us just for first time. Welcome to ask your questions about parasitoids. This is my email. And let's start to watch insects under the microscope, and not only under the microscope. First of all, you can say we did solve question about rearing parasitoids, but sometimes you can collect parasitoids just in the nature using just very simple butterfly net, or we can say sweeping net. So I am doing this kind of sweeping, just pushing just my butterfly net one way, another way. My butterfly net can be called as a parasitoid net, because I'm sweeping one way, left, right, and many small insects and big insects can escape quickly from a net, but small insects can remain inside the net. But then I will be coming to the special emergence traps, which I use for separation trap. Maybe one day I can show it precisely under the high magnification, but I will do this separate presentation for separate stream. Today this is just, I remind you, just this summertime, but we are collecting insects in the field using even this very simple way. Here I am collecting insects in Pirogova Architectural Museum, ethnographic museum near the Kyiv, where we have just huge area with old ethnographical museum with old houses which were built in Ukraine in the last centuries. So very wonderful museum with buildings and with churches. So everyone who is coming to Kyiv in the spring, summertime should visit this museum to enjoy this museum under the open air. Okay, and what we should do with these insects? These insects should be mounted, not pinned, because my parasitoid's pretty small, 2 millimeters size, the biggest up to 10 millimeters size. So they must be glued with glue on small paper cards, and we attached with label, small piece of paper with name of country, village, region, in landscape, and date of collecting, and name of collector. So all you see different papers, different cards, different insects collected in different places by different people. So but they are together because they are belonging probably to one same species, and it's stored in one big box with collection. And insects not very big, so 2 millimeters, 2-3 millimeters size of family here Torimidae, the egg parasitoids of a praying mantis, belonging to the genus Podagrion. So they're not so attractive like butterflies, by the way, for sure, because parasitoids quite small, but under the high magnification of microscope, we can observe them together here as a big size insects under the microscope. Even small insects can be observed as a quite big. You can see on the right side, this is egg mass, egg of mantis. Podagrion splendens, identification label. So, and what happens if you can observe living organism? Yes, you see here. This is another genus of a family Torimidae, is the same previously. Podagrion, Torimidae, and this one is Torimus. So under the big magnification, head, antennae, and black color eyes, composed eye, first pair of legs, and what she's doing, this is a female, she's just sinking, licking the honey. Yes, all the insects, especially parasitoids, must be fed with water or with water with sugar, or you need to give just a drop of honey to feed them before observations, because under the light and during the heat in a hot place, parasitoids will die very quickly. So for observations, it's important to give a little bit food for insects, as here we see this very nice parasitoid Torimus is licking the honey. Even can make a magnification, of big magnification for a head. And you see here legs. I want to show you. Yeah, and this is how it looks like. You see, it was head about 2 millimeters and total the size about 5 millimeters, with very long stick on the top of the body, and it is called, you already know, ovipositor, special structure for oviposition for laying eggs. And this is gall-forming parasitoid, which is laying eggs inside plants, inside plant galls. So swollen pieces of plants on stems, and so this Torimus is parasitizing another Hymenoptera, gall making Hymenoptera of family Cynipidae. So that's why this female must use so long ovipositor to penetrate very hard stem of gall, and to penetrate this gall to search for the larva of its host. So special ovipositor, and so very precise specialization of parasitoid. So you see, this insect pretty small, about 5 millimeters size, but even big parasitoids like this one have something similar, but this parasitoid is a huge one. This parasitoid belonging to the family Ichneumonidae. This is genus Megarrhiza, which is sitting on the bark of tree, on the bark of a birch, dead stem of a birch, so no leaves. So that's why somebody already was living inside the wood, inside the dead wood, and before the whole tree was cut, still insects will be eating the wood, and this interesting parasitoid is searching for the host, horntail wasp of a family Tenthredinidae sawfly, very special sawfly, Tenthredinidae family, which is larva of which is living deeply inside the bark of tree, under the bark of tree. That's why this very long syringe or just long stick, which is used by this insect, is used just for drilling, deeply drilling inside the wood, to making hard efforts to you see the top of a body, the top of abdomen is moving and making hard job, just pumping different parts of ovipositor and using it like a, not only like a syringe to put inside something, but also like a drill, like a screwdriver, to make a very careful drilling of wood and cork, and then finding the larva under the bark and laying eggs inside the body of host. And this is all ovipositor, so long ovipositor, longer, for some species longer than body of the whole parasitoid, this Ichneumoniid wasp. So this insect was sitting carefully, and so I used the camera to observe for maybe 5-10 minutes to catch this moment, this unique moment. And also some birds can come. So that's why this insect is absolutely helpless. They are not stinging, they are not biting, they're very soft, very nice meal for birds. That's why sometimes on some wood it's possible to find just only very long sticks, only ovipositors, because some birds come in and feeding on these nice parasitoids, nice, very sweet food for birds. But if we're alone, if we have very nice mimicry to be not visible, dark wings, just some spots on the body, so we're not very well visible. Only if you know how to find them on the bark, you need to observe them carefully near the dead trees, and my God, you can be lucky to find such wonderful parasitoid. Sometimes we are flying very quickly. If we are not drilling the wood, we're just crawling on the bark of tree, searching for precisely for the place, clicking by antenna on the bark of tree, searching for the host. And after finding it, female is stopping, curving just very long ovipositor, and put it inside and drilling and making her hard job. And after that, if you want to collect this wonderful specimen, you need only just to take it by fingers and keep it for a few minutes, because female cannot take it off the tree, because it's still inside the tree. You should wait a few minutes before female will take it off. So I did the same. So I was standing near the tree, just it touched to the tree, touched to this parasitoid before collecting it, because I could not take it off without damage of female. I needed to keep it in my fingers very carefully, and just wait several minutes before female took off ovipositor out of the wood. Don't forget about subscribing to my channel. This reminding you about it. So welcome if you're first visitor. But we can continue and see another insects. We can see this is insect of a close relative. This is a Braconidae family under the microscope. Nice dark wings with stigma, with pterostigma, and also with ovipositor, but not very long. This is sample under the microscope and collected and stored in alcohol. So you can collect insects and keep them dry in collection, or partially they can be collected in water, in ethanol, and kept in a sample before the study in ethanol or alcohol, 70% alcohol. So here this is a wing of another parasitoid, another Braconidae, I guess, because this is a very special venation. And yes, some cells of venation. We can see antennae, first pair of legs, and the second pair of legs, and just the body. Well, so here I'm trying to make smaller and bigger magnification to show the size of insects. Yes, here this group of Braconidae parasitoids, different species. So different colors, different shape of body, different shape of wings and venations for sure. This is probably parasitoid of aphids, Aphididae, I guess. Also here, this is a Braconidae wasp. Braconidae parasitic wasp. Majority, the parasitoids of stigma, pterostigma, such as swollen part, enlarged part of a vein. And here, very short ovipositor. Yeah. Here much longer ovipositor. This stick on the top of abdomen, this is ovipositor. And another species of a family Ichneumonidae, you see, not the same species as was sitting on the bark of tree, but close relative, Ichneumonidae relative. Here I show that this parasitoid not stinging, not biting, so don't worry about the behavior. Just very carefully you can just even play in fingers. She is flying very well, but in kitchen there is no way to escape, and you can play a little bit before releasing it to the nature, by the way. So if you play, you can take some videos and then you can release it back to the nature. And next parasitoid of the same family, Braconidae. Already this is a short video of behavior in the laboratory. So here, this is a white part in the center. So this is caterpillar and black bodies of parasitoids of a genus Habrabracon. Bigger magnification. So just moveless caterpillar and many parasitoids, many females. These are females of a genus Habrabracon incubator, and this is Indian mealworm of a species Plodia interpunctella. And what's happening here? Here just it's happening, each female trying to lay eggs, but it's not always successfully, because before laying eggs, some females need to feed itself, so the drilling the caterpillar, which is already parasitized. So caterpillar was already attacked by one female, and after that becoming moveless. Female can insert ovipositor, and after that caterpillar becoming paralyzed, moveless. So, and also female of Habrabracon feeding on extract from this hole in the body of caterpillar and becoming fed, and the body is enlarged, so very big one. So that's why they're becoming very happy, very well fed and sitting nearby. And after feeding, after a short period after feeding, females try to lay eggs already. They are not immediately lay eggs. In some cases, if eggs already possible to lay, so females will lay. But in many cases, firstly they just feeding on the body of caterpillar. So that's why they are not so active, not crowding and not fighting between them. But in many cases, if one female laying eggs, after that next female is coming, just penetrating the previous eggs, killing them, and laying new eggs on the same body of caterpillar. So there is kind of competition. If population is pretty high, too much dense, so you see here many females. So there is a competition for the same food, for same meal. That's why one female of parasitoid can kill the progeny of another female, can kill with ovipositor eggs of another female. So that's why you see here on the left part of screen, female is just leaking liquid from the body of caterpillar. So we're not moving so much. So we're not moving with ovipositor. Just leaking the hemolymph, which is very nutritious, so important for germination of eggs, inside the body of female. And next parasitoid can come to our screen of a, probably you have seen it, but I'm not sure. Yes, no. These are parasitoids, egg parasitoids, family Platygastridae or Telenomine, were separated in one box after sorting. So you see here we have a short antennae, short swollen rounded abdomen, short thorax, and very simple venation of wings. So these are Platygastridae, egg parasitoids, probably parasitoids of eggs, some Lepidoptera, but they have a wide variety of hosts. So Platygastridae, egg parasitoids, already collected in ethanol, so stored here in the sample, a glass jar in ethanol, 70% ethanol. The best way to collect insects, to keep them dry, and second way, to keep them in ethanol for preservation, either for DNA study or for taxonomical study before making microscopical slides. And last but not the least, this is a parasitoid of a family Trichogrammatidae, my family which I am studying. So and here you can see the title. This is a male of a species Trichogramma dendrolemi. Why it is male? Because you can see on the right side the antennae with hairs, elongated long antennae with hairs, and also you cannot see here later on the body genitalia of female but genitalia of male. So short body and no wings for this species Trichogramma dendrolemi. There are some variations, there are some males with wings and some males without wings. If population is too dense, some males appearing without wings. So this is a male without wings. And don't forget that this parasitoid is rather small, very small I would say, because the size, you cannot imagine, this is the size of this insect, just 0.5 millimeter, just half of millimeter size of this parasitoid. Parasitoid of eggs of different night moths and deadly butterflies. Even this, despite the size of 0.5 millimeter, the structure are the same in the order Hymenoptera, like a small wasp, like a small parasitoid, with antennae, head, thorax, abdomen, legs, and all necessary tissues and structures. So, but this parasitoid, egg parasitoid, not the smallest one, don't forget about it. Smallest will be the parasitoid of genus Megaphragma. I showed it already in some of my videos. Watch my videos some days before. In genus Megaphragma, the size of parasitoid 0.2 millimeters, 0.3 millimeters, 1/3 part of millimeter, and Megaphragma is parasitoid of thrips, tiny sucking insects, which are well spread in many areas, but Megaphragma is still known only from semi-tropical area and desert area, so more or less we're associated with warm territories, with warm areas and landscapes where thrips are distributed. So you can search for these parasitoids and it can make discovery if you can find them. And also can collect these trichogramma species for identification in your country. And I am interested to study them taxonomically. Maybe we can find new species of trichogramma in your country. Never known new species trichogramma in your city, in your region, as I said, in your country, because the size of them is pretty small, 0.5 millimeter, difficult to collect and difficult to study. And if you collect them and keep them in some samples, in small samples, in ethanol, it's easiest way to collect trichogramma and small parasitoids with brush, with tiny brush. If insects just will rear it from the host, the best way to take insects with brush and put them inside glass jars or Eppendorf plastic jars, plastic tubes with inside ethanol, inside 70% ethanol. So, and put inside label with data of country, city, village, landscape, date of collection, and maybe possible host, plant host, or just name of host, insect host, butterfly host, and close it toughly, because ethanol can be evaporated from Eppendorf tube. But you can keep them before identification in refrigerator under the -10, -20 degree. And you can send these samples for identification to our laboratory, to Institute of Zoology of National Academy of Sciences in Kyiv, Ukraine. Maybe this species which you can collect in your country will be new species of trichogramma never described before in scientific literature, and it can be useful for a description of new species for the science. It can be done together with you as a scientific publication. You can be co-author, or you can be just person whom I can express my acknowledgement inside the scientific paper. If you like to be co-author, welcome. Just collect material carefully, find them original place and original insect, and maybe you can discover originally new species of insect, egg parasitoid, or maybe new place of distribution. So maybe some species will be not new, but never found in your country, in your distributional area, and it will be also good material for scientific paper or publication in scientific journal, like this journal, Journal of Natural History. Why not? So a very prestigious journal, very prestigious publication about parasitoids. Here, this journal was devoted to precisely to parasitoids. So, welcome for collaboration. Why not? Collaboration is always good if you have careful observations and careful rearing of parasitoids and careful storage of parasitoids inside glass tubes, inside plastic tubes. So you can always send them to our laboratory in Kyiv, Institute for Zoology of National Academy of Sciences in Kyiv, the capital of Ukraine. So don't forget about my email. If you are lazy to search for my email in the internet, but you can always find this email on some sites where I give my contacts. And don't forget, if you are interested in to be a sponsor of our channel, welcome to Patreon page. Patreon page link is given under this video. Patreon page, so you can send couple dollars for a cup of coffee and for support of my channel as a feedback for my efforts in distribution of scientific knowledge and proposing some kind of collaboration between scientists. So try to collect parasitoids and welcome for cooperation. We can talk about cooperation in details later, but you know now my contact email and always can write your questions. And under the video, if you have Gmail account, so it's allowed to you to write comments under this video. Write your comments, write your questions. Welcome to Patreon page. Press like. Don't forget, Ukraine is finally fighting for freedom and independence. Ukraine is forever. Good luck and see you soon on my channel. Bye bye. Good luck. See you soon on my channel, on my new streams. Good luck. Ukraine is forever.